Welcome, everyone. We're glad that you've joined us to celebrate Stability Day 2025. Today, we'll recognize stability stakeholders for their role in the stability function, determining the shelf lives and optimum storage conditions for medical products that are saving, preserving, or improving the lives of people around the world. We'll be answering your stability-related questions, sharing some challenges and trivia, and introducing you to a variety of stability suppliers. Helping to answer your questions today, we have a panel assembled in an alphabetical order. Jeff Carr, who is a consultant, and he's actually based in Canada. And right now, he and a valiant technician are up on his roof trying to restore his internet. So he's not with us for the moment, but we hope that he can join us soon. Jeff Cook, Chief Operations Officer, Parameter Generation and Control. Maria Krish, Research Fellow, Physical Sciences, Freethink Technologies. Chris Latos, Manager of Stability Services at Hollister Incorporated. Myself, the PSDG Facilitator and Editor at stabilityhub.com. And joining us afternoon time, Laura Pack, Head of CMC Quantitative Sciences at Moderna. And then Walter Routh, our founder and CEO of Sagana Quality Consulting. And Walter, can you tell us why we celebrate Stability Day on February 19th?
Oh, what a good question. So Arrhenius, the father of stability, and the Arrhenius equation, I'm sure you've all heard of that, was born on February 19th, 1859, so 166 years ago. So this is our third consecutive Stability Day that we've celebrated, and it focused, it's the first one, I think, that we've been able to coincide directly with his birthday. And I think next year we'll be fortunate to do that as well. So that's what we're doing, celebrating his birthday today, but often President's Day or Fridays. We don't like to set meetings on Fridays. So if it's a Tuesday, Wednesday, or Thursday, we'll probably celebrate directly on the 19th. Otherwise, anyways, just exciting for everybody to be here.
Thank you, Walter. We closed last year's Stability Day programming spontaneously imagining that a Stability Day parade was concluding. And when that conclusion happened, we imagined that Svante Arrhenius came by on the last photo of the parade, much like Santa does at the Macy's parade. When he came into view, we were done with the program. In evaluating that program, we concluded that a special day of celebration really did deserve a parade, even if virtual, and that the elements of a parade might be as entertaining as possible to introduce our participants to the various organizations that our panelists represent and open up the door to your questions as well in each of their areas of specifications. With appreciation to Walter for merging his intelligence with artificial kind to produce our 2025 virtual parade, we'll get underway with the Stability Hub Float. So honoring the Pharmaceutical Stability Discussion Group founded in 1988, which later opened its online presence as Stability Hub in around 2016. The float depicts the very first PSDG meeting, complete with all 7 of the folks gathered and a single conference table and flip charts to record the proceedings. Disregard the fact that they have laptops. Artificial intelligence is sometimes not as intelligent as we would like. And that brings us to the first question of the day. Where can we find stability information? I just showed you my favorite way, the stabilityhub.com, which brings you opportunities to view publications, legal requirements from the various guidances, as well as any other contact you might want to have with discussion groups. You can also see your suppliers, organizations, and find jobs at the stabilityhub.com. So that's my favorite way, but you can also learn stability information by attending stability seminars, getting handbooks. Organization websites are great to visit, things like APPS, Kennex, PDA, USP, and ISPE, among others. And becoming an active member of the stability community will get you easy access to most of these. And Walter, do you have a bit of Stabilitana for us?
Well, besides the fact that we were founded in 1988, another thing regarding PSDG is the most famous member that we've had was none other than Wolfgang Grimm, the father of ICH. Actually, you can tell us what year that was, if you might remember.
That's a good question. It was about 5 years in, so it was probably the early 90s, right after ICH got going. Wolfgang decided that the world needed to harmonize a little bit, so he had no idea that he was going to end up with ICH. but he called a lot of stability leaders together, I believe in Heidelberg, possibly Frankfurt, and they got together and had a discussion, and ICH was born from that. So that was great. He happened to be at the host company we were meeting at that year, and they dragged him in and said, "You need to listen and see how some of the stability folks that you've been gathering over and overseas are now meeting in the U.S." And so we greatly appreciated his participation. Okay, let's move along to our second float of the day. And this is the group from Parameter Generation and Control. They brought a large part of their staff to ride on the float, and that's Black Mountain in the background. And today is so cold it looks like Black Mountain turned blue. But we'll let Jeff Cook tell us a little bit about Parameter Generation and Control.
Yes, hello everyone. My name is Jeffrey Cook. I am the Chief of Operations here at Parameter in beautiful Black Mountain, North Carolina. It is actually a bit snowy on the ground outside right now, so the mountain is accurate. Parameter Generation and Control has been manufacturing high quality storage equipment such as cabinets, reach-in chambers, walk-in rooms for over 47 years. We pride ourselves on the level of constancy that we can achieve through our two-temperature dew point humidity control method that utilizes a spray chamber, the dew point of the water bath, and a reheat process. This process eliminates the need for highly purified water, steam injection, or the utility infrastructure that comes along with both of those things. Our customers span industries from pharmaceutical to paper, packaging, and textiles, to lab equipment and standard manufacturing environments such as clean rooms. Whether it's one of our standard product offerings for a stability storage room or a custom design built and based on your workspace and application, we have vast experience in tackling the nuances of humidity controlled environments.
Thank you, Jeff. And we have a question for you. And I'll point out to the audience that please use your chat feature if you have questions. We're organized this meeting primarily for you. We have a few questions in advance to get us started, but we're glad for answering your questions. We'll give those priority as soon as we see them and can match them up with one of our panelists. In the meantime, for Jeff, what are some of the common issues you encounter with people that are installing stability chambers?
Well, the most common issue we encounter is probably just lack of preventative maintenance. A lot of our units will be put into interstitial spaces or mechanical rooms. Depending on the application and the use of the equipment, the preventative maintenance might not be needed terribly often. But because of that, our machines can easily be forgotten about, and they will run 24/7 for years on years. And just over time, a bit of maintenance is needed. And through personnel changes and as studies roll over, the machinery can kind of be forgotten about and neglected. But the machines do, in their various applications, typically run 24/7. So if you think of your car running 24/7, at some point it does help to give it a little tender loving care. So the PM should be considered, it should be planned to prevent downtime in the midst of a study. Depending on your application, it may be needed just once a year. It may be needed every 6 months or even quarterly. We recommend that at least once a year you check on your machine, change the filters, the UV bulbs, etc. Another frequent issue we encounter is going to be trying to operate the unit outside of its intended range. As studies roll over from year to year, the unit can be applied to a room that's too large for its intent. Room insulation can break down, or the unit can be applied to a room that's not very well insulated. The ducting to and from the machine and the distribution of the air throughout the chamber can have a significant impact on its performance. So just as a unit has, we pride ourselves on our units lasting upwards of 20 years, sometimes even 40 years. Over that course of time, if the unit was bought to do, say, 5 to 25 C, inevitably the question arises, well, can it do 35? So we find often that machines will push the envelope of the machine. You mentioned install as well. Because our machine is typically capital equipment, it ties into a lot of other infrastructure and is often the last chain in the last link in a chain of events that needs to happen in order for our machine to be turned on. You have water utilities, power utilities. Often it's a construction site with a whole bunch going on. And so a lot of things have to go exactly right. And sometimes we even tune the machines on site after all the construction is done to make them work optimally.
Great, thank you, Chris, or Jeff. Get my attendees mixed up here.
Perfectly
So
all right.
we have time for a question, and one of the ones that came to mind was what's a stability story actually. That what's the most unusual thing that's ever happened to a stability chamber? And in my memory, I think it was when a hurricane came through and ran up the East Coast. A lot of people down south are used to the hurricanes by these days, but in New Jersey, where this company was located, they were not, and they had located their stability area in the basement of a building at the foot of a hill, on which was a parking lot. So no place for the water to go but down. And they went on and discovered after the hurricane was over, they came back into the plant that all the stability chambers had been flooded out. And not only were so watertight that they didn't admit water into the chamber, but that it caused them to float up to where the ceiling level was because of the water level. And by the time they got done, the chambers were no longer functional. The products were scrambled, and they decided to move the stability area to a different location. So weird things happen in the stability world, and we just want to bring a few of those to you from time to time. Let's see, another question has come through. Do the units have the capability to integrate with LIMS software? That would be for Jeff.
Yes. So depending on the generation of unit you have, we have recently rolled out a new controls platform that will integrate with any sort of building management system, SCADA. I'm not terribly familiar with LMS, but the way that we have built our new controller is that it will communicate via Modbus TCP variables or protocol up to 35 variables of just all of the workings of the machine, the air temperature set point and actual, humidity temperature set point and actual, the outputs of all of the variables and motors and fan speed. The software that we have built is a read only, so you cannot actually manage the unit remotely through a BMS. That was intentional. A lot of our customers really do not want the risk of our machines to be operated externally because a lot of them are Part 11 compliant machines. They are very controlled environments. So the new controller does have the remote connectivity option. At the moment, we have disabled it for our standard product, but there are various projects that we're working on with the intention to expand and build that connectivity and control in. The
Great,
new
good to
controls
know
platform.
as
Go ahead.
well. Both Parameter Generation Control and Novatech have sponsored webinars on the Stability Hub site. And it's interesting because that this question pertains a lot to what Novatech can do with some of their programs, and I'm sure some of their competitors can do it as well. And that's to pick up the signals and reports from people, and through the digitalization process, integrate it with some of the other systems so that you can read those things. And I think that's a good call about not allowing operation from externally, because sooner or later, somebody's going to be able to hack in and drive your chambers nuts. So it's a really good situation to make sure that you avoid that. Okay,
Exactly, John.
thank you. Let's move on to our third parade unit. This is Jeff Carr Consulting's band. And Jeff has managed to place his instruments in the middle of the brass section through the miracle of AI. So really appreciate Walter capturing a really good likeness of Jeff. He probably doesn't look like this up on his roof right at the moment, but Jeff's consulting business has apply applications to the laboratory, especially contract laboratories, test method development, anything on the chemistry line applied to pharmaceutical development. stability testing, and training for all of the above. So we appreciate Jeff, and hopefully he joins us a little bit later on to field some of the questions as well. And Walter, anything else in the line of stability trivia that you would like to show us?
Well, one interesting tidbit I came across is the fact that if you have a water deluge in your chamber, similar to what you were just describing in your story, um, it won't necessarily cause a significant acceleration in the moisture that is ingressed into the norm, other than the normal pH or RH of the room. Meaning you didn't kill your studies, is what I'm saying. You may need to dry them off just to keep looking pretty, but you actually won't. The water contact with the surface of the bottle actually, because of the hydrophilic nature of most bottles, are hydrophobic. It's being right up against the edge. There is no vapor pressure to push the individual molecules through the material. So you end up with the surface tension barrier right at the edge of the bottle. So essentially, you don't end up with any additional moisture entering your product as a result of a flood, for example, except of course if it can get up through the cap and just make a mess of everything, obviously, and you don't want it. It's out of control, obviously, but still, good news is that it doesn't necessarily mean your study got killed.
Hey, thank you. We have a question. In general, if a stability study is being outsourced to a CDMO and the client company decides that they want to conduct some additional testing on samples in that study, are there ways available to allow this to be done without compromising the study? I think Walter and I could take that one, and I'll start it saying that, yeah, I think CDMOs are glad to oblige, but the big limiting factor is, did you give them a surplus of samples and put it in your contract that you could make those kind of arrangements so that they're not totally caught off guard when you make those kind of requests? And Walter, you have some experience along those lines. Anything to add to that?
Sorry, I was busy trying to admit people. Could you repeat the question?
No problem. It was whether or not when you send your samples to a CDMO then and give them a protocol to follow, and then later on you think of a couple other tests you might want to do a one-time sort of thing on. How does that rattle the CDMO? Can they handle that?
Yeah, well, it all comes down to what kind of change control you have with that CDMO. I mean, if you've got something in place where they have a process for you to implement your changes, there shouldn't actually be too much rattling going on. They accepted the study with that in mind. But you need to have that. That's one of the things that auditors really have looked at for me in the past as well, is to see what kind of change control we have with our contractors and such.
Yeah, I would imagine if you just willy-nilly sent that in and they complied without a whole lot of paperwork and the change control, that would really draw some eyebrow raising when the auditors came through as to what kind of operation was being conducted in both shops. Any of the panelists are welcome to join in on that if you have any additional remarks on that. If not, I have one for Maria. And that's how is accelerated predictive stability, which is the specialty of the company you represent, Freethink Technologies, used at different parts of the product development cycle?
Should I do a little Freethink intro first?
No, we're going to save that for a little bit, but.
So, yeah, so accelerated predictive stability studies are basically short-term experimental studies which are used to model shelf life in packaging. So we have temperature and humidity as main variables, doing experiment on your product, and then you're modeling shelf life for that product only. So it's not a first principles calculation, it's very empirically based. And so it can be used at all sorts of different parts of the product development cycle. A lot of companies use these kinds of studies to screen either choosing an API, choosing an early formulation to make sure that whatever's chosen has a reasonable stability. This has a lot of advantages over sort of just rank order because you may have all of them that are acceptable or none of them are acceptable. Later in the product development cycle, these kinds of studies are used by a lot of companies to get into the clinic faster, as well as to target packaging for clinical supplies so they don't have to be doing packaging screening or start off in a packaging that starts failing. Later on, they're included in a lot of different ways as part of a package, a registration package, sometimes for supporting changes late stage, sometimes for understanding excursions. Often even in post-approval, they're used for post-approval changes to sort of demonstrate that a change might be a minor change. So lots of different uses for models. Ultimately, these models are Arrhenius-based models, so they're very appropriate for talking about today. They're moisture-modified Arrhenius models. And so basically understanding how a product behaves is useful at various different points of the cycle.
Great, thank you. And also, could you comment a little bit about regulatory acceptance at the various stages and how that might have been changing over the years to where it is today?
Yeah, so these kinds of models have been sort of increasingly used. And again, because the science is sort of, basically Arrhenius-based behavior. So there's been an increasing use, and a lot of the early companies using these kinds of models tried very hard to put them into a lot of regulatory applications, partly to get the regulatory authorities to be comfortable with them, understand what they meant. So I think how much they're leaned on by themselves versus in conjunction with more traditional data really depends on what part of the development cycle you're in. So for some situations, they're actually used as standalone. Things like, API, starting material, shelf life. Often, phase 1 clinical shelf life, that can be used as a model only. Often what companies do is they'll have a model, and then they'll also have more traditional stability that's set up and may be tested only upon request. Obviously, as you get later on in the stage, you move towards more traditional stability as the centerpiece of an application, and these models are usually used as more supporting information to understand changes, enhance the package, handle excursions, things like that.
Great, well, thank you. Let's see, I think the Novatech International float has arrived with all hands on deck, each not unexpectedly wrapped around a laptop. And Novatech is not joined us yet today. I think they might have some of the same problems up in Canada that Jeff Carr is having. But Novatech is one of the sponsors of Stability Hub, and we appreciate that. They do a lot of work with digitalization, and they've written some articles on the difference between being digitized and digitalized. Tongue twisters for us to wrestle with these days, but there is a difference in the meaning. Digitized means simply that the data is in a digital format, and digitalization has a lot to do with how accessible that data is for pulling together for a common report. And going out on a limb and going to Chris Latos with a question that we had in assembling some of your regular systems, such as a LIMS or maybe a stability tracking or those kind of things. Is it important to make sure that you are lining up the possibility for them to be digitalized later on? In other words, will they have to have hooks that will enable a higher system to reach down and pull out the data? Is that something you might be able to answer for us?
Yeah, I mean, a lot of times you have a lot of different systems that don't necessarily talk to each other. So you've got, information maybe in a limb system. You may have a separate limb system that you do your stability studies on. We have, SAP. Some companies have product lifecycle management. So you have, all these different, warehouses of data. So, you 4 or 5, multiple data warehouses. So yeah, ultimately, if you can tie it into one system, you've got one source of truth that way. What happens sometimes is you have kind of redundant information in two systems. Sometimes things get changed in one system, but they don't get updated in the other system, and that can create problems. So yeah, ultimately, if you can have everything tied into one major system, a single source of truth. Of course, that's a big ask. But yeah, ideally, that's what you would want to do.
Yeah, maybe the cooperation between your IT department and your user groups in establishing all kinds of user requirements could establish that up front early enough so that the supplier can put those kinds of hooks and arrangements in. I know in the good old days, if you had one stability system you were dismantling and then implementing another one, there was no way other than hand entry of each single piece of information into the new system. And hopefully those days are disappearing, that we can now not lose the flexibility of our old systems when we come in with the new system as well. Okay, Walter, thinking of olden days, you have a picture of a certain product from olden days that we could talk about? I'm challenging Walter's flexibility here.
Yeah, that's let's stop sharing this and pull it up just a sec.
Yeah, I forgot the intricacies of doing that. While he's doing that, let me remind you that we have over 50 people registered for this meeting, a quadrupling of our past records, and we really appreciate that. And you must have some questions represented out there. We don't want you to be shy. Any question is fair game. You can ask us about having backup stability chambers, secondary monitoring systems, what to do with a product that you manufacture 6 months before you put it on stability, what's time zero, how to handle submissions to Brazil. All kinds of questions are just lurking in those brains of yours, and we'd like to entertain them. So feel free to flood us with those kind of questions. In the meantime, Walter has shown us a bottle that's sitting on the shelf behind me in my picture, although you can't see it. And is it possible to blow this up a little bit more? Maybe not. It's Cheracol with codeine from way back when. And we wanted to ask what kind of stability problems you see with that. Well, that's great for the label. So let's focus on that for a minute. Does anybody see anything that's a little bit different or missing from that label? And feel free to type it in if you want us to do that. See, we have an exempt narcotic that gets away with just being recorded in the pharmacy notebook. The measurements of the ingredients are in grains and mins, which are drops and subsets of grams. You'll notice there's no storage conditions noted there. You could store that any way you want.
Yeah.
And hopefully the bottle takes care of the light, but I don't think it takes care of the heat. And then Walter, if you could just go up to the top of that bottle a little bit.
Also, a few, Sarah and Jeff are noting that there's no expiration date or any date or lot number.
Yeah, exactly. Perfectly fine answers there. And the top, well, have you seen one of those? I think I last saw it on the wine bottle I opened up last night. So maybe not too antiquated, but for pharmaceuticals, not so good.
Yeah, talk about leachables. How do you test for the various kinds of cork trees that were used and the different species? I don't know.
Yeah. No NDC number from Natasha. Thank you for that contribution.
Mm-hmm.
So there's a bunch of stuff there. So we've come a long ways over the years for this. Oh, you'll notice that one of the ingredients is wild cherry syrup. And just as to let you know how far we've come, when I first started my pharmaceutical career in pharmacy school, we were required to make wild cherry syrup from the bark. It was an NF product, so we made it according to the national formulary. And I haven't met anybody else who's done that over the years. I think we've gotten a little bit more modern in our manufacturing methods. Okay, let's see. We do have some questions that are coming in. Let's go up to the top. Recommendations for stability inventory management. Yes, we do that. I'm going to open it up to the panel in just a minute. I think Chris Latos, this might be your bailiwick. I've done lots of different methods for stability inventory, and none of them are convenient. They usually take a lot of time. Sometimes it involves pulling the samples out of the chambers, and that's not good for maintaining their their temperature, especially if it's frozen. So it's problematic that way. Sometimes people do a rotating thing chamber by chamber, or else they tackle the whole thing with all hands on deck for a couple days a year. We did that once during an FDA inspection because they found that our inventory was off. So we pulled everybody we could. We stayed up all night, recounted every single thing in the chambers, and we introduced 53 new errors in doing that, probably because we were sleepless. So there's a problem with a lot of those things. I like to recommend these days that you inventory every time you do a pull for just what you're pulling. And then in the course of a year, in theory, you've inventoried everything and you keep it updated that way. So Chris, I've given you a little bit of time to think about inventory management, maybe from the higher levels and what kind of programs are out there that might be helpful.
Yeah, so I will give a plug shout out to Novatech. So that we have the Novatech stability limbs. So the inventory management is built right into that. There's barcodes that we use, so that's a a really good way to track what chamber a sample is in and so forth. So when you remove a sample, you just simply scan the sample out. System works good, but yeah, you still need to do some type of physical inventory. I think like many people, when a study ends, 3 months after the study ends, you walk in a chamber for something and you find some samples that have been laying around that never got discarded after a study ended. But yeah, barcode labels, scanning, that's really, helped our stability program.
Okay, great. And we can also put these questions on the website and have anybody else that wants to give some answers participate in those kind of discussions. We hope to capture a lot of things off of this meeting and move them forward onto the website for an even wider audience. Let me go down here. Status of the updated stability ICH guidance documents. Unfortunately, we don't have a panel member that's participating directly in that, to my knowledge. It is coming along. The reports I've heard is that they're making great progress. There's a committee that's doing that, and there are subcommittees in various industry groups and companies themselves that feed into that. So if you can access one of those people, they'll give you a great deal of information. And will they include a guidance for cell gene therapy? I think that that's under consideration, but I don't want to say that for sure. So we're going to take this question and do some research for you, and we will post it on the site to see what we've learned about the cell gene therapy guidance, as well as where the ICH guidance is now for the complete reorg. For those that don't know, they're consolidating a lot of the current ICH stability guidances into one and clarifying a lot of things as they go. So it'll be very interesting to see when that comes out. I would guess they're still a year or so away, but don't quote me on that. So we'll be back to you more on the website with that one. Oh, and somebody's got an answer for us. There was a final concept paper in 2022 for targeted revision of the guidance. Is there an update? Okay, sorry, that's about as far as we've gotten before. When placebo is a different formulation from the drug product buffer, is it required to perform a separate stability study from the drug product, or any chance to leverage data from the drug product? Let's see. That Walter, would you know about that, or Maria?
I wouldn't, so I'll defer to Maria, although I would think when we were talking about a clinical stability study here, I would think, and that's, but I do not know the answer.
Yeah, sorry, unfortunately, that's not something that we're covering either. So
Okay, then it falls back on me to to give an answer from my experience. A placebo is a different formulation from the buffer. I would say you do have to perform a separate stability study on it. That's just my opinion, having faced a few auditors here or there. If it's the same or very close to it, then I think leveraging is an absolute way to go. So if you want to make a case that it's different but not terribly different, put together your best justification. Make sure that your quality assurance and your regulatory folks are on board with that. And if they're feeling confident, then by all means, ask the question of the regulatory authorities. You might be pleasantly surprised and find out that it's okay with them. But it's dangerous to proceed and assume that you don't need a separate study without making those questions known to the regulatory authorities. How do you manage annual commitment lots and study start when lots are released later in the year? Samples are ready, samples ready into the next year. You have to use the year in which they're manufactured to do that. And study start when lots are released. It's always tricky when they get manufactured in December. So we've always recommended that you always try to schedule those annual commitment lots for November or earlier in order to get them placed on stability. It's pretty messy when you go with something that was manufactured in December and you put it on in January. It starts to raise some eyebrows amongst the regulatory folks. So I would say managing is to make sure that you try to get them early on. And if you can't, then perhaps just declare nothing was made in that year, as long as that's the truth, and push them into the next year. Another question on chamber alarms delay time and chamber alarms response time. Number of either 30 minutes or 60 minutes set in our monitoring systems before we receive alarm emails. For response time to chamber alarms, it varies, ranging from a few hours to 24 hours to the next business day. Wondering what others in the industry are doing and their justifications. Boy, Hong, you are a busy woman these days. I would say that delays are part of the normal SOPs. 30 minutes is pretty good. 60 minutes, you would want to have justification, like it being a room temperature alarm sort of thing. So not much is happening for the delay. So that's the alert. And then how fast your response comes would also be based on the kind of things that you have already set in motion for getting people on site and set up. So you could send security people in just to give you a more accurate picture of what's going on. You already know there's an alarm, but they could tell you whether or not they're seeing smoke or water on the floor or anything unusual. So it's good to have eyes right there if possible, and you can communicate with them from home. But it's also good. A lot of companies, if you have enough personnel available, is have a response team set up. So at any given hour, you know who's going to respond. They know the procedures, and they will get directly to the site, take over control, and either move samples, turn things off, or intervene. And again, based on the risk factors there. Let's hold there for a second and continue on just a little bit, and we'll come back to your questions. Let's get over to the next.
I'd like to address a little bit on that as well, though. Just
go ahead. Sure.
Yeah. One recommendation I would make is that you define various lengths of excursions or times that you might have. In this case, you could call it, we did in the past have called it a routine time or a routine excursion. So those were defined as when for maintenance or just door openings and various things. And they were also given a limit of, I'm not sure, 15, 30 minutes. So then you could, once you define those intervals, then in your procedure, you can say, a delay is built, beyond a routine excursion, for example, and kind of builds your justification into that.
Okay, thank you. I'll take the other one next, the next one very quickly. It's about what to do with the OTC guidances. A lot of times you can refer back to some of the more formal ones for NDAs, ANDAs. But there is an OTC organization out there. We just added their recommendations to our website under the publications section. So take a look at that. They do have things that are a little bit less stringent, and they seem to have the blessing of the FDA. They go very carefully with their organization. So take a look at that publication, and you'll be able to see what's going on with the OTCs. And if you still have more questions about that, by all means, pop it into the discussion forum on the site, and we'll try to make some more comments on that. Let's hold on the next one for a moment and go to the next parade unit. I hear a wheelie mighty roar heralding the approach of the Hollister motorcycle unit. Chris Latos, how does Hollister roll these days?
Thanks,
Sorry for the corniness.
Yeah, no, I love it. So welcome everyone. My name is Chris Latos, and I'm the Stability Services Manager at Hollister, and we're a medical device manufacturer. We're located in the far north Chicago suburbs. We manufacture disposable medical devices, class 1 and class 2. Some of our products are sterile. The majority of them aren't. So our big product lines have to do with continence care, primarily intermittent catheters. So these are for patients that have to self catheterize in order to urinate, spinal cord injured patients, wheelchair bound, and so forth. And then our other major product line has to do with ostomy products. So people that have, severe Crohn's disease or colorectal cancer, they have to get part of the intestine removed. And then a opening is created, and waste comes out of this opening that's generally in the abdomen. So our products actually capture the waste that comes out of the body. So pouches, adhesives to fix the pouch against the body and so forth. Those are ostomy products. Let's see. I'm a bit of an anomaly. I've actually been at my company now for 33 years this October. So only second job I've ever had. So yeah. So yeah, is
Congrats.
there anybody have any questions on medical device stability?
We had one who submitted, what are some differences between drug stability program and a medical device stability program?
Yeah, so there's, and I'm talking strictly medical device. Drug device combination products are a whole different ball game. But for strict medical devices, it's a lot different than, drug product stability. A lot of the stability indicating tests are very well defined in the drug industry. Assay, impurity, maybe dissolution, some of those types of tests. Medical devices span a whole gamut. Again, the products I just talked about that my company makes, but anything from a tongue depressor, our products through pacemakers and things like that. So it's a multitude of different medical devices. So there's no, it's very difficult sometimes to determine what exactly is a stability indicating test. So that's a major challenge, and I think a major difference. With our products, again, there's no assay, there's no, active ingredient. So a lot of our tests have to do with physical testing. So we're looking at, again, these are plastic or polymeric materials, components. So we look at things such as, tensile strengths of seals, primarily heat seals. Do the seals degrade as the product ages, especially for a sterile product. With our catheters, it's very important lubricity because it's being inserted in urethra. So we want to make sure that the lubricity, the coefficient of friction is the measurement we perform, that doesn't change drastically as the product ages. So I think that's a big difference is determining the appropriate stability indicating test for the device. Then again, when you involve, sterilization, packaging is very important. So you have these interactions of, the product with the packaging, the sterilization modality, be it, gamma radiation or E-beam, what impact that's going to have on the device as well as the packaging. And there's not really well-established guidance for medical device stability. There's a FDA guidance that was written in 1991 and has never been updated. There's some other bits and pieces that you can find. So primarily we rely on ICH guidance, but a lot of that doesn't necessarily apply to our medical devices.
There's a follow-up here. Are the conditions that are used for medical devices tested at ICH conditions as well?
Yeah, that's a good question. Yeah, so typically we do ICH conditions, so we'll do the 2560 for real time. Accelerated is where it varies a bit. We'll do 4075 as the typical accelerated condition, but based on our device, what polymeric components, what thermal properties, melting behavior of those, we can go as high as 60 degrees C for accelerated aging. When we run a study at that high of a temperature, as a contingency, we always run 40, just in case, something catastrophic happens at 60, we can at least rely on our 40 degrees C accelerated data.
Okay, thanks.
Along
And
that, I would think
yeah.
you would want to not add humidity. Like you would want to dry. Is it possible you'd want to dry it at a higher temperature as opposed to moisture? It seems like the drying impact would be greater to your
Yeah,
plastic.
it really depends on the packaging. Like some of our packaging constructions are impervious to moisture, so humidity, it doesn't matter what humidity you set the chamber at. By default, we just default to the 75 RH. But you're right, different packaging, yeah, maybe more realistic to run it at a 20% RH.
Thank you.
Okay, we have a question about what is the start date of the annual stability study? Is it the date samples are placed in the chamber or date manufactured? Would like to benchmark what others do and reasoning behind it. I've seen it both. So here's a technical challenge for us. Walter, can we do a poll like hand raising in the chat room?
Actually, if people just if you know how to raise your hand, it's oh shoot, I can't remember what.
It's under React, I think,
Yeah,
at the bottom.
so if you could just raise your hand there, we'll be able to.
The first thing we would ask in the poll was how many people can answer the question if they know when the date samples are placed in the chamber, start the study, or is it the date of manufacture? So if you can answer that question, if you could raise your hand electronically there.
Oh, that's nice. It puts the hands raised in the top. So
Do we have a feature that counts the hands?
yeah, it's called me.
Oh, okay. I won't distract you then.
So I'm seeing 10 so far.
Okay, let's call 10 our baseline. And then out of those 10, and you can put your hands down now so we don't confuse it with anything else. I think Walter can also take the hands away. How many people there would use the date in the chamber of those 5? Typically, PSDG polling, the 20 would say yes.
Yeah, so we've got 8 so far. Looks like 9. So we got 9 and
9 out of 10. So let's assume that the other person would use the date of manufacture, and people want to raise their hand and unmute to discuss. You're welcome to do that. I have some comments, but you've been hearing my voice all morning, so I'll wait a second and see if anybody else would like to comment on the reason why they do the date and chamber. Or
Hi,
the one
John. This is
person.
Denise. I'm the one that asked the question, and I
Sure.
asked it in the Stability Hub, and I've asked it over the years because I've seen it both ways. So, hello, fellow stabilitarians. We started out when I first started my career was the date in the chamber. Then there was a shift, probably about 10 years ago, where I was at Novartis, where we used the data manufacturer, and the reasoning was for the annual they were already at considered a long-term condition. From the data manufacturer prior to being placed on stability. Now I'm seeing it at some other employment where they want to use the date in the chamber. So now you're testing like way beyond expiry at times, depending on when you place the sample in the chamber from the data manufacturer. So just curious as to what other places are doing. And I know you put a time point at expiry. But just curious as to what others are doing and if they've had any issues with auditors with what they've chosen.
Well, I'd love to answer as the data manufacturer side. I think there weren't anyone except me, unless someone did raise their hand. So we did for 20 years, we were doing data manufacture exclusively. And the rationale was that the study or the product prior to being placed in the chamber was being handled in the warehouse at the normal manufacturing and the normal supply chain conditions. I grant the negative side of that is the stability commitment says months at a particular condition, months at 25, 60. But typically the warehouses were at 30 degrees or around there. So our argument was that it certainly wasn't being stored in any less stringent condition than our chambers prior to placing on stability. The other significant argument, or at least our rationale for doing that, was that exactly what you mentioned at the end there, the ability to not test beyond expiry by placing it on the date of manufacture, the expiration interval that you insert coincides with the last interval typically, or at least usually within 30 days or so. So there's actually a significant time savings, or cost savings, less testing beyond expiry, and you also have savings from less unnecessary investigations for failures beyond expiry. As far as the question of inspections, we were never questioned on it. We were, I have to admit, a little worried about the possibility of questions, but we never got that question from an auditor. We did have a white paper ready, if necessary, to justify it and explain our rationale. So it worked really well for us, I have to say.
Yeah, and I agree, Walter. Sorry to interject again. Yeah, because I agree with the data manufacturer. I'm just at a new position, and they're kind of wanting to use so I'm kind of trying to gather data on why we should use a data manufacturer. I agree on everything you said. That's my logical reasoning, but it's not really working. So they want to do the date in chamber, and now we're testing way beyond, and we're investigating things that are way beyond expiry, et cetera. So just maybe we could hear from the other point of people who use it based on data in chamber and your reasoning behind that as well.
One more thing I want to say real quick is just one thing we implemented very importantly at the end was to expedite the time from date of manufacture to date in chamber to reduce that and hopefully reduce the risk we might have of an auditor's questioning how much time out of the committed conditions.
While people are thinking about their answers, if they want to unmute and speak, I'll just read Crystal Hill's response. For her particular situation, it differs based on molecular type. At her job, small molecules are date and chamber, and large molecules are date of manufacturing. And to the comments that Walter and Denise gave, I would say that the great principle here is whatever path you take, have some scientific justification. Make sure you cover all your bases that if you're counting the time of manufacturing and waiting for the first study to actually hit the chamber, then how the units are stored and controlled and secured. Cover all those bases, and I think you could take it either direction that you would like to do that. So we'll open it up. Any other comments on that question?
Well, the absence of any, I'm just going to point out that refrigerated or frozen samples would be very easy to justify. It's more the room temperature, 30-degree long-term storage that might be harder to justify and prove that you had controlled conditions prior to stability.
Mm-hmm. Well, thank you for that, Walter. And now we'll go back to our parade. And I think what's coming up is a bunch of folks who really get into the Stability Day spirit. Freethink Technologies is out of the box and looking to generate some data ASAP. And Maria can, wait a minute, that's not them. This happens to be Lighthouse. So we can talk about Lighthouse for a second. Lighthouse Instruments, who drove their float right around the Freethink float to get in front, will be reprimanded severely at the end of this parade. But they make instrumentation that helps with headspace, particulate counts, and those kind of things. They were not able to send a representative, so we don't have any particular questions for them, but we just did want to give them a mention and recognize the fact that they've been very supportive in the Stability Hub realm. So now we do have, in the next one, the folks that are celebrating Stability Day at Freethink Technologies. And Maria, what can you tell us about FreeThink? And also some of the programs that you have coming up are quite exciting, the science of stability and those kind of things, if you could comment on that.
Thank you very much, John. Yeah, so from FreeThink Technologies, so we're a company that tries to do sort of creative approaches to understanding stability and formulation. So we're probably best known for our software offering, which is called ASAP Prime. It's currently the only commercially available program that's used for both planning and modeling this accelerated predictive stability data. So these are empirical experimental measurements, making a model, and then predicting shelf life and packaging. And they are Arrhenius based, so it's very appropriate for Stability Day. In addition to our software offering, though, which is fairly widespread at this point, we also work as a contract research organization. So for companies who might not want to put in the infrastructure to do these studies themselves, we perform a lot of these, what we call them ASAP studies, to make stability models. We also do formulation development, especially sort of complex formulations, ranking formulations, and some analytical method development. As John alluded to, we have, an interest in sort of promoting new and interesting science around stability. And so for many years, we've either sponsored or co-sponsored a conference on stability. And that is something that alternates between the US and Europe every other year. This is a US year, so hopefully there may be of interest to some of the folks at the meeting today. So it's actually going to be in downtown Philadelphia in May, specifically May 19th through 21st. And that's called the Science of Stability Conference. This year, for the first time, we're also sponsoring a conference that's focused more on larger products and biological product stability. That's going to be in October in North Carolina. So if you're interested in either of these, we have a website for the conferences called scienceofstability.com. I think John has often posted them in the event section for Stability Hub when they come up close as well. But we'd love to see people from different companies and perspectives giving presentations or just attending. So thank you.
Great. And we had a question about the goal of ASAP.
Yeah.
What's the goal of an ASAP study and how would one go about that? And people are welcome to ask questions as well for following up on this.
Yep. So an ASAP study is fundamentally, you're trying to make an Arrhenius model with a modification for moisture dependence. So this is a short-term experimental study on a particular product. And the major goal is to be able to make a model of shelf life in packaging. So the model has two main variables. It has temperature, it's the Arrhenius part, and humidity. And so, you can basically predict the behavior at an arbitrary storage temperature. And then with some inputs about what your packaging is doing, to understand how that changes with packaging, because that's modulating the internal relative humidity. So, people use these for all sorts of things. They use them for screening different packaging, screening different formulations, screening APIs, establishing clinical shelf life expectations, supporting excursion behavior, lots of different questions.
And are there any products that are not yet in the realm of ASAP? I know that Ken Waterman is continually pushing the envelope and looking at new applications.
Yeah.
There are some things that don't fit the model just yet.
Absolutely. So yeah, and that's something that, as a company, we're sort of internally do a lot of research to try and make sure we understand where the edges are. So fundamentally, this is a question of what follows Arrhenius kinetics. And we're using an ISO conversion approach, so it's somewhat model independent. But most chemical reactions are going to follow Arrhenius kinetics. There are a few physical stability areas which also model well. So the most widely used one, I think, has been dissolution, disintegration, things like that. There are some physical processes which just have fundamentally different things going on. So things like sedimentation, that's going to be completely different processes. But generally, things that are sort of activated barrier processes are fundamentally chemical, like a color change, which is really coming from a chemical. That's what tends to model well. The other thing is you can have things that model well, but have too big an error bar. These are statistical models. So generally, in order to study something, we need an analytical method that has a error bar, if you compare from the starting point to the spec limit, of less than 20% of that gap. If it's bigger than 20%, it doesn't mean that the fundamental process can't model, but you'll get a model that basically, it's like a Captain Obvious thing. It's true, but it doesn't give you useful information. So you need kind of a good analytical method and Arrhenius behavior.
Okay, thank you. So let's return to the viewing stand of the parade. There's no horsing around with this next unit. The Moderna equestrian group, where every horse is fully vaccinated, has trotted into view. And Moderna is represented by Laura Pack, who is head of CMC Quantitative Sciences at Moderna. And the quantitative part there and the science part is just a tip of the iceberg when it comes to Laura. She is the most knowledgeable person regarding stability statistics that I've run into in my entire career. So we welcome her and ask her to tell us a little bit about Moderna, and if you want to skew it towards statistics or CMC quantitative sciences, that's fine.
Well, thanks, John. Thanks for having me, and thanks for the kind words. So Laura Pack here as, I guess, representing CMC Quantitative Sciences. So my team covers pretty much everything in CMC that makes data. That's how I like to describe our scope of responsibility. And we help turn data into decisions. So stability is a large part of what we work on. And, I started my career in stability, which helped me become a very effective statistician when it comes to analyzing stability data. So over here at Moderna, we do work with an interesting drug product label claim that's 3 storage temperatures in sequence. So that's a new thing for me in my career. And then, of course, we also support plenty of other things besides stability, but that's always going to be my first love. I'm a stabilitarian at heart.
Can you comment a little bit more about the sequential storage? That's new for me as well.
Yeah, I think it's a vaccine thing, John, and I became familiar. Actually, there's a couple of WHO guidelines that tell us how to do the math. So we have, as many of you may remember from the vaccination campaign, the vaccine long-term storage condition is frozen at -20, and then there's a refrigerated storage period, and then also kind of a patient convenience storage period at 25°C to allow operations at pharmacies and such. But once you take it out of its previous temperature, you don't go back. It's not total time at this or that temperature and going back. It's colder to warmer, like that. And so we do kind of a piecewise regression to support that.
Is that something that's guidance driven, or can we expect to see that expand in the industry, or we're just not aware of it happening and it's been around for a while?
It's interesting because there are WHO guidance documents. It's Technical Report Series 999 Annex 5 that describes how to do the math. There's an older version, Technical Report Series 962 Annex 3 maybe, but we like to reference the later one. And it describes kind of an equation that's a piecewise regression with a propagation of error to tell us kind of how to calculate that total change that you expect based on those 3 storage temperatures, and then add some uncertainty in your slope estimates and your method variability to kind of figure out how much change you can allow over the shelf life. It also includes a paradigm for 2-tiered specifications where you have to guarantee that you're not going to release either, for an attribute that decreases over time, you're not going to release below a certain level to make sure you stay within specification through the end of shelf life, or you can flip that for an attribute that increases over time.
Are there any publications out there that describe what you just described yourself?
Besides the regulatory guidelines, I am not sure that there are, let me back that up. I'm sure there are some articles, but I don't have them to quote right off the cuff. I can send you some. I will write myself an action.
Yeah, that'd be great. No rush on that, but it'd be nice if
Yeah.
we posted it on Stability Hub.
Yeah.
Folks can access. Would be
Yeah.
fantastic.
Yeah. Of course.
Okay. We cannot let you leave without you telling us a little bit about what your opinion would be, or what are the elements of success in establishing a good stability statistics program, just at a high level. What are we missing that we end up with 483s and those kind of things when it comes to our statistics programs? I know I've had a statistician directly on my staff for 5 golden years, and it was heaven. And then I lost him, and we had to train some new people or send it out to a group which had some wonderful folks and also had some terrible folks. So what do you see as the best thing that we could be doing?
Yeah, as far as 483s, I think I don't see a lot of actual observations. We generally have, as long as we've got our validated methods and you're following your stability guidelines and you've got your procedures in place and you're following them, I don't see 483s. What I see more of are questions, right, on dossiers and how are you supporting your claims? Are you doing analytical method transfer correctly? Are you following, are you establishing your shelf life claim appropriately? What I would love to see as a statistician always is a few more extra decimal places in the data that we get from our partners. And I have this, I guess it's a soapbox. I'll call it a soapbox because there's a dichotomy between what's the reportable precision in your results. That you have certainty because you've validated the method. So maybe my spec is in a whole number, and so I only get results in a whole number. It's especially difficult when I'm getting results from a CMO. That's a little bit of a higher hurdle. But I like to consider, statistics are a calculation, right? And so we wouldn't round our numbers as we go through a calculation. And so I would always recommend that we add at least 2 decimal places beyond the reportable precision as we're going through our statistical calculations. There's a good paper on chunky data that talks about how overly rounded results can make a reliable process seem unreliable. And so for stability, that really hits us hard because we're establishing our shelf life based on a confidence interval on the mean. And if we're using overly rounded data, that confidence interval can be wider than it needs to be, which can artificially shorten our shelf lives, for example. It can also prevent us from seeing signals in control charts and in lot release data and things like that. So that's my true desire. We'd be able to make better decisions with better data.
Great. Thank you very much, Val, for that. And we did have a follow-up question for you. Do you mind sharing with us what software you use to do the piecewise regression? And if you can't, that's okay too.
Well, so I allow my analysts some flexibility to use whatever tool they're most comfortable with. I think it's more important that you're following the same statistical analysis method, that you're using the right model, and verifying your model assumptions. So as a quantitative science department, we use JUMP to teach our non-statisticians how to do analysis because it's quite versatile. It does almost everything, at least well enough, right? You can design experiments, you can do model fits and make graphs, etc. Internally, for more experienced statisticians, we use R or SAS mostly. And then, there are, of course, you all know perhaps better than I do that stability databases often will automate your standard ICH Q1E analysis. I know ICHQ1 is being revised to incorporate some more advanced modeling techniques, right? Talking about maybe mixed models, Bayesian approaches, accelerated stability, Arrhenius approaches, and some of these things that JUMP may not do as readily, or you may have to, it's maybe a little more manual approach. And these kinds of analyses are also not automated in a stability database package. There are some really good books out there that will walk you through how to do the statistical analysis, either in just the model fit or also in JUMP. And I'll send you a link to a couple of those books that are actually free PDF downloads.
That would be fantastic. Thank you so much.
Of course.
I missed a question a little bit earlier I want to go back to, and that's we have a recommendation for frequency of stability chamber mapping. I have recommendation, but I'm going to be quiet for a minute and see what other people would like to say about that. Recommendation for the frequency of stability chamber mapping, which is slightly different than a requalification, but sometimes included in it.
Anyone, if you want to unmute yourself and respond, that'd be great. Happy to hear a variety of answers here.
I can contribute a little bit to this conversation just from my experience in the service department here at Parameter. I like to see mapping more often than most of our customers generally do. I would say 3 years, every 3 years you should probably map. That is not from the testing side of it, that's more the equipment side of it. We do see blower wheels get dirty, airflow disruptions, ductwork will get pinched as contractors are walking on top of stability chambers and things like that. If a sensor is being recalibrated annually or even semi-annually, if the sensor placement isn't in a good airstream, that can very much have an effect on the uniformity throughout the room. So I think I see a lot of customers of ours trying to push the envelope based on the cost of requalification or mapping. And I generally like to see the mapping happen more often so we can adjust fan speeds and make sure that the product's placed well on the shelves in relation to the sensor and everything.
That's very interesting. If I could ask a follow-up from you on that, because it's neat to have you answer it as a chamber expert and manufacturer. Have you seen a significant shift in a chamber's mapping or the way it's behaving after, say, replacement of a compressor or a fan, a major part that might be different? Have you seen a shift in the mapping?
That's a really good question, Walter. I would have to see a mapping, just before the replacement and recently after the replacement to really know. But I do know that when we replace compressors, as much as they are a standard manufactured piece of equipment, you're also going to want to tune the different refrigeration valves to get the uniformity of temperature across the coil. That will then result in the uniformity of the temperature through the ductwork and in your room. Even the fans, a lot of the fan manufacturers will put small weight balancers on the different fins of the fans just to make sure that the fan spins smoothly and doesn't get off balance. Depending on the age of the fan that has been replaced, over the course of that fan's life, our fans are variable speed through the controller, and it could be that as a fan ages, we want to up the speed to maintain the same CFM. If you go ahead and replace a fan that's been in operation for 10 years, it might be the new fan is more economical or uses less power, and that same fan speed setting alters your air distribution across your room. So very hard to prove, but it could certainly have an effect.
Thank you.
A few more comments have come in on that. One, Chris Latos, we perform chamber mapping requalification every 2 years. Crystal Hill says, in my previous position, we remapped every year. And I would comment as that being very typical. We've taken lots of polls in PSDG meetings, And it ranged from every year for people who are super cautious. 2 years is written in some guidance someplace, but if a few people do that. 3 is the most common one with industry. That's probably best practice that you could make a case for. And other people do it as necessary because they're monitoring their chambers. And as soon as they see some variation or deviation, and as well as in the guidance, it says if you change your load patterns, you need to remap at that point as well. So there's lots of reasons to justify why you remap whenever you do. The worst case is you don't remap at all and you don't have good justification to do that. So people should pay attention to that, but have a really good reason why you're remapping and requalifying from time to time to do that. And I think that almost all those positions are defensible with some good data. Okay, back to the parade route. Do I hear music? The Sagana Consulting Marching Band is here. There they are, 2,000 strong. And Sagana is Walter Routh's company. And it's obvious to see that Sagana expects 2025 to be a banner year just by reading those banners in the back. So Walter, what's playing at Sagana?
As you mentioned, I started in 2022 when I retired from AbbVie. I had 30 years of stability experience there, and just carrying that on. Sagana Quality Consulting, we really specializes in stability consulting. I've got a lot of stability experience, and I have so many years were spent working with management to reduce our costs, and trying to carry that over to anyone else who needs to help reducing costs, increasing efficiencies, and at the same time not affecting compliance, or even I could have a lot of experience helping people to be audit ready, and just looking at what areas they need to watch for in their audit. So that's the experience. It's you can reach me at Sagana QC.com or email walter.ralph@SaganaQC. Just look forward to hearing from anybody if you have some questions.
Thank you. I do have a question that came through. Where would you see the biggest areas in the stability function that people could actually enact some cost savings? From time to time, we see that happening. People want to know. I participated in a lean stability program one year, and we came up with some ideas, but those ideas have changed over the years as to what's the safe way to go about that and what's the most effective ways. Do you have any ideas as to what people can look to?
Yeah, already just from some of the questions, the amount of time spent inventorying or checking inventory can, in my opinion, from a lot of companies, be reduced. The amount of time spent mapping. I agree with Jeff that 3 years seems ideal. If you're doing it more than 3, unless you've got some really old chambers that are breaking down a lot, maybe, but another area that's a low-hanging fruit to change. Those kind of things could be just switched in the blink of an eye, just change your procedures and such. But then there's so many others, like reducing intervals or not testing beyond expiry, things like that. So those are a few quick examples. Yeah.
Okay. Monitoring for questions. I noticed there was anyone interested in taking a poll regarding the remapping intervals? We could do a quick one of that. And I'm thinking we don't need to necessarily do a baseline, but we could add up the ones who respond. And watching the hands over here, we'll put Walter back to work as our chief counter. How many people chambers every year? So it's a 1-year basis. Put your hand up electronically if you would do that for 1 year. We have to do some scrolling, but I'm not seeing any hands.
There's one. You said how many people map once a year, right?
Once a year.
Yeah.
Okay, then take that hand down if you would, and put it up for 2 years.
Did you say 2?
2 years, yes.
Okay.
And you got those counted. I see another one just went up. Okay, you got the count?
Yep.
Okay, and take all those hands away and 3 years. Every 3 years for revamping. We all set with that? More than 3 years. And I'll give you some more options in just a moment. Anyone more than 3 years as their program? 4 or 5, whatever. Don't see any. Then again, I haven't scrolled. So 0?
Yeah.
Okay. Never. How many people don't do it at all? And in a minute, I'll ask how many do it as needed. So something they've not considered, never remapped. And we will not be sending these reports to the FDA. Nor has the FDA registered for this meeting. So no nevers. How many people strictly on an as needed basis? Different events occur. Okay. Did I leave any other possibility out? Okay, I think we're good.
Okay.
So I'm starting to hear music. No, wait, we heard music in the last one, didn't we? Sagana. So we'll hold off for a minute. I have time for a commercial regarding Stability Hub. And it means I could bring up the following points. If you haven't signed up for Stability Hub's newsletter, in other words, if you came in through LinkedIn or some other advertising for this meeting, you can sign up for the newsletter. It comes out once a month, has all kinds of events and articles and things like that to point you to, as well as finding jobs or making discussion, just like we've had here, only on paper. You can go to stabilityhub.com and complete the brief drop-down form, and it will get you signed up. If you haven't already signed up yourself, please tell colleagues about us. That helps us to meet more people, and it's also very encouraging to our sponsors that more people are seeing them as a community player as well. Our next webinar is in collaboration with Parameter Generation and Control, and it's scheduled for noon on Thursday, March 6th, and it's focused on stability chambers, generating user requirements, how to select chambers, and that is another in the series on chambers that Parameter is doing. There'll be office hours webinars that are very similar to this, only minus the parade, in May and October to take your stability questions. In the meantime, we welcome your questions on the Stability Forum. All are welcome there, and we try to get an answer back to you as soon as possible. Stability Hub is powered by volunteers. If you would like to assist in an archival or research capacity, or have plans for a summer intern that could spend a couple of hours a week becoming more familiar with all aspects of the stability function, we could keep folks like that busy along those lines. So we'd love for anybody else that had some free time. It's not rocket science. We're looking to see how many articles we put out on subject X or Y and when they were, so we don't repeat ourselves, or we see what's missing and we can focus in on new articles and new areas to bring to you. So those are the kind of things that we're thinking about. We have time for some more questions. How often should you open door or power recovery studies? How often should you do the open door or power recovery studies? So I have an answer as a user for that, but I'll let Jeff go right to that as the manufacturer if you have some recommendations.
Well, shoot, John, I was reading the offerings on the Stability Hub website. My man, you sold me on your on your ad. Can you re-ask the question, please?
Yeah. How often should you do open door or power recovery studies? And if you want to think about that for a second, I'll say as a user, we basically did it when the chamber was purchased and was part of our installation qualifications, operation qualifications, especially to do that to see what they recover. But they possibly, I could see that being done in the PQ as well. So any recommendations from a manufacturing point of view, or are these strictly regulatory driven?
Oh, certainly, John. So I am familiar with the open door and the power down studies. My familiarity is most often when upon initial qualification after the install. That is where us as the product provider are most exposed to that. Oftentimes, the requalifications are handled externally to Parameter since we're just the manufacturer. So I'm most familiar with it happening just upon initial startup. I do know that the machines now will give you notification when there is any sort of power fault, and you can build the notification of the door openings in there. I have very often been flagged of suspicious activity or suspicions that there is something wrong with the equipment when actually the door is just being opened erroneously by a maintenance person in the middle of the night or someone who's just curious to see what's inside the room.
Mhm.
Cool.
Okay, any other comments on the recovery, or if somebody does it a little bit differently? Okay.
I think what I'm hearing is you really don't need to expect to do an open door or power test at during requalification intervals, especially not often. That's important to note.
Yes, and it serves a major purpose when you're trying to evaluate. You suddenly discover a door's been open for a couple hours, and then you close it. So it's good to know how long it's going to take to come back up in your evaluation of any excursions that have happened. Power recovery is going to last a little bit longer when the power goes off because there's no disturbance in the conditions unless you open the door. So all handy things to do, especially if you're trying to put together a report to come up with some kind of evaluation of your event. Let's see, for Jeff, another one. What are some of the most unusual conditions you've seen for a stability chamber or room, and what was it for?
Oh, we've seen temperature and humidity chambers used for everything you can imagine. Breeding ticks to study Lyme disease, putting snakes into hibernation states for antivenom studies, um, having a stability chamber full of animals, testing how efficient you can make animal feed by, evaluating the manure. Spark-proof chambers for volatile chemicals and explosives is a fun one, with 4-foot concrete walls and a tiny little stability room.
I think we're going to stick to the stability realm. That sounds like pretty stinky jobs for some other people here and there. Well, thank you for that input. I think we need to take a look at the parade route one more time here. So rolling into view is the Svante Arrhenius float. And Svante is waving farewell to us as we close out our program today. We're getting very close to our limit. So I wanted to thank the folks that are participating in the panel, for those of you who are attending as well. We'd love to get your feedback. Let you know what you liked about this program, what you might find frustrating, ways to improve it. We wrestled with the length of it. Last time we did it was 2 hours, but we had it starting at the top of the hour and ending at the top of the hour. And we realized a lot of people were racing from conference rooms that ended just at the point we were beginning. So we moved it 15 minutes back and ended 15 minutes early and came out with 90 minutes. If we were overwhelmed with questions, we discussed the possibility of adding on an extra half hour to an hour, depending on what you're going to do. This format is very close to what you used to experience in person in a stability discussion group meeting. So if you've enjoyed this format, we're always looking to see how many people might be interested in meeting in person for a couple of days. We really had some fantastic meetings that people love, but lately there's not been the support from management to send people to those kind of meetings. Recording of this program will be accessible in the events section on our website for a while, and major questions will be posted in our discussion forum. So that's available. And that basically does it for us. We wish everyone a happy Stability Day. Thank you for joining us, and we'll see you in the future.
Thank you.